Hello, everybody. I just wanted to go through the first couple of slides that we went through today. For whatever reason, some sessions I explain things incredibly well, or at least I think so. And in other sessions, I feel like I don't do such a good job, simply because like I just get fumbled up in my mind. And basically, with this recording, I just want to go over briefly what we talked about today for the foundations of biochemistry, simply because we are the things that are going to be holding you, um, like things that you're going to be using to help interpret other pathways. I just want to make sure you have a good foundation in it. Okay, so I'm just going to briefly go through all of the terms that most of you, that all of you, hopefully, those of you who attended class today have heard. So metabolism, just all the chemical reactions that occur within a living cellular organism. Metabolism is the breaking down of molecules down to small precursors. So remember, we use that analogy of the Legos. Okay, so we have a house and we want to, get just the individual Legos back, and we break down the house, right? The Lego house. Anabolism is the building up of things. So let's say we broke down the house of Legos. With anabolism, what we do is we use those individual Legos to create something else, maybe like a car or a boat. Next, we have cellular respiration, which again is just the metabolism of the creation of energy from foodstuffs, right? So cellular respiration is just a specific kind of metabolism. It's just that metabolism associated with getting energy from food material. So really quick, let's go into oxidation and reduction. So we talked about how oxidation is the loss of electrons and how to recognize oxidation. Like rather than just the loss of electrons, you can visualize it with the oxidation with the gaining of bonds to hydrogen, or the loss of hydrogen atoms to a molecule, or the loss of electrons. And for reduction, we said is the gaining of electrons. So in reduction, way to visualize it is just the loss of bonds to oxygen, or the gaining of hydrogen atoms, right? We use the example of O2, water molecule. Remember, O2 looks like this. Moving on to H2O, right? We said that this would be H2O. It's a very ugly H2O, but it's an H2O nonetheless. So here we have two bonds to oxygen, like these bonds are attached to each other. These oxygens are attached to each other with two bonds. And if we were to move the arrow in this direction, for example, towards this product, what we find is that the oxygen is now bound to two hydrogens, which means it has been reduced. And as we mentioned before, the oxidation reactions and the reduction reactions, they're always paired, right? So in this direction, rather than it being reduced, here we have bonds to hydrogen, and we move this direction, we have two bonds to oxygen, which means that we lost bonds to hydrogen. If we lose bonds to hydrogens, that means that we're oxidizing, right? So that's reduction and oxidation. And recall that with catabolism, they're breaking down of things that usually is paired up with oxidation, right? The oxidation is just extracting electrons from a compound, extracting those electrons. Typically, those electrons are used to build something, or those electrons are used to, like, for example, facilitate the electron transport chain to create ATP. So the electrons are just being extracted from a molecule, and that's usually what we find with catabolism. When we're breaking things down, we're going to oxidize it. That's not to say that in catabolic reactions, there's not going to be a single reduction. There most likely will be a type of reduction in a catabolic overall reaction, but mostly the process is going to be facilitated by oxidation, the extracting of electrons from substrates. And for that reason, when you oxidize and you oxidize and you oxidize, the product of cellular respiration is CO2 and water. And those things are very oxidized. Like this carbon is double bonded to your oxygen here. And for that reason, oh god damn it, I wasn't screen sharing. Well, I think that's fine. We were just going over terms anyways. So here I was just showing the oxygen, how it's double bonded here, and we want to go from this double bond to the bond to the hydrogens. So if we instead of having a bond to oxygen, we have bonds to hydrogen. So if we gained bonds to hydrogens, that means we've been reduced. And redox reactions are always paired. So if in this direction we have a reduction because we have new bonds to hydrogen, we go in the opposite direction. Here we have just bonds to oxygen, which means that you have an oxidation rather. Okay, yeah. So with catabolism, you usually have the oxidation, and with anabolism, the main reaction you're going to be using is going to be the reduction reaction, which is going to be reducing things reduction is gaining of electrons, so you're just going to be giving electrons to substrates for them to be become your target molecule. Let's say you're trying to make something for your muscle group, so reducing something so you can give it to your muscle group. You're making a target molecule, or just reducing something to store those electrons in a storage molecule. For example, fatty acids. Like fatty acids are a long carbon chain, and they're a really good way to store electrons because fatty acids are made up of long chains as such, and those long chains can be broken down into twos, like two carbons. And those two carbons can become acetyl-CoAs. Remember, we talked about how acetyl-CoAs can really provide energy for the cell. Okay, so after talking about oxidation and after talking about reduction in terms of catabolism and anabolism, let's just quickly revisit Gibbs free energy. So we said Gibbs free energy is a way to determine if a reaction is going to be spontaneous or not. We talked about how in a reaction mechanism, if the reactants had higher energy than the products, that means that they're going to go down energy towards the products. And a change in G, remember, is a products minus reactants. And so if our products had lower energy than the reactants, then in this here equation, P would be a smaller number than R. A smaller number minus a bigger number is going to be a negative. And when you have a negative change in G, we have a spontaneous reaction. On the other hand, if we have a positive change in G, if we had a positive change in G, then we have a non-spontaneous reaction. We had the example of the ball on the hill, right? So this ball going down will occur spontaneously. This would be a negative change in G. Or the ball trying to go back up, that's not going to happen. It's not going to happen spontaneously. It's going to be a positive change in G. Okay, so that's Gibbs free energy. That's the way to look at reactions, right? As we mentioned before, for Gibbs free energy, that there are going to be some reactions that are non-spontaneous. For example, if you're trying to build something up, something's not going to be randomly built by itself. Like something has to facilitate that. And that's why cellular respiration is really important. It's because we have these reactions that allow unfavorable reactions to occur, right? So let's say, for example, we have an unfavorable reaction that has a positive change in G. Right, this is the reason why we eat food. We eat food so we can create, for example, ATP. And ATP can be used for this purpose. For example, like I say, A has a very positive change in G. Let's say it's a Gibbs free energy is 8, positive 8. And then B, we have a negative change in G, which is 9. G, which is 9, right? So what's cool about Gibbs free energies is that you can add reactions together. So you can add A and B reactions together to form a product. And if you add positive 8 and you add it to negative 9, you get a negative 1. So overall, although this reaction A, was initially non-spontaneous, due to the spontaneous nature of this reaction, and we add those together, the overall reaction is going to occur, right? So that's why we eat in the first place, right? We eat so that we can get molecules like ATP that can act as our spontaneous reaction that can couple to the non-spontaneous reaction for us to do stuff, right? Like if we want to move around, it takes up ATP. That ATP is going to be hydrolyzed into ADP. As we discussed before, ATP looks like this, aka adenine attached to 3 phosphates, right? If we hydrolyze it, we're going to have a lot of energy released as heat. And that heat is going to facilitate whatever reaction that happens to be thermodynamically unspontaneous, right? So that's why we eat, so we can get ATP, and ATP can drive the reactions in our body, right? It's cellular respiration. ATP or whatever reaction that happens to be spontaneous and coupled with a non-spontaneous reaction, that's called reaction coupling. Okay, in terms of photosynthesis, the next term that are on our list, we're going to skip fermentation for now. That'll be in the next video. So for photosynthesis, that's just to cover our bases in terms of like, okay, where does this whole concept of cellular respiration come from? Like, where does it start? Well, it starts with photosynthesis when plants take light energy and forms molecules that we as things that eat other things, not things that create our own energy. It creates molecules that we can eat and get energy from. So photosynthesis begins the whole thing by the plant, the photoautotroph. So photo means you get light as your source of energy. Auto means that you made it yourself. So the plant will get light energy, create something like a sugar. And then us chemoheterotrophs Chemo means we get our energy from chemicals, like for example, ATP. We can get energy from ATP. Hetero, because we don't make it by ourselves, we get from eating other things. And troph just means that we exist, right? So that's just covering the basis of photosynthesis. Like cellular respiration begins when photosynthesis in a plant occurs, and then something eats that plant, and we eat that something that eats that plant, right? That's how cellular respiration occurs, and then we have our pathways in our body to make our food into energy. Okay, so now just to talk about the different ways that you can create ATP. So ATP can be created by either substrate level phosphorylation or oxidative phosphorylation. So substrate level phosphorylation is, as it sounds, a substrate or a molecule is going to donate a phosphate group to ADP. So adenine diphosphate is going to create ATP, right? Adenine triphosphate. That's just substrate level phosphorylation. Direct transfer of a phosphate group from a substrate to ADP. Oxidative phosphorylation, on the other hand, is when you have the electron transport chain, aka one of the cellular respiration pathways that we're going to be discussing. When we have the electron transport chain, create our ATP. Remember, the electron transport chain isn't a single molecule; it's a chain of reactions that that allows ATP to be produced. So we'll get more into that mechanism in a little bit. Okay, coenzyme is just the enzyme. Let's say the enzyme is doing a thing, is doing a reaction. However, some enzymes, it can't do the reaction by itself. It needs a coenzyme. It needs a assistant to help it with a reaction, right? Let's say, for example, our reaction was a oxidation reduction reaction. Let's say the enzyme was a dehydrogenase. Well, our coenzyme that's going to be paired with the dehydrogenase would be most likely NADH or FADH, because these are electron carriers, as we explained before, right? The electron carrier, what they do is, let's say we oxidize a compound, where the oxidation is what? It's the removal of electrons. Where are those electrons going to go, right? They're just going to go into free space. Like, what's the point of oxidizing if we do that? So no, the coenzyme, like NADH or FADH, you're going to catch those electrons and then use those electrons to create ATP by putting those electrons into the electron transport chain, right? Those are electron carriers. Okay, continue forward. So important enzymes that we talked about: hydrolase, breaking things with a water molecule, like proteases, like for example, a peptide bond is going to be, if you add a water molecule, the hydrogen will go to one of the molecules and one amino acid, and the OH group will go to a different amino acid, and then you'll have a separation, right? This hydrolysis is the breaking up with water. Isomerase is just the making an isomer of a product. For example, let's just say we have this OH group here. And an isomer, what would do is that everything has the same chemical formula, but you just have a isomer of what you initially start off with. A kinase is just the movement of a phosphate group from a high energy carrier. Like, let's say ATP is a high energy carrier. The kinase just takes the phosphate from the ATP and it puts it onto another compound. The compound, typically, if it becomes phosphorylated, it just means that it's going to be activated. Or if it's not going to be activated, it's just going to be part of a cascade. So let's say molecule A is phosphorylated, and then a kinase phosphorylates molecule B, and that phosphate group just goes to C, D, and just continues down the cascade until it reaches its target. Right? Okay, so phosphatase, if kinase adds something to, if it adds phosphate to a molecule, phosphatase would just be its antithesis and removes a phosphate from the molecule. AKA, typically it's going to be deactivating a molecule if it's removing the phosphate. That's phosphatase. So I kind of skipped over to enzymes. So oxidoreductase, for example, dehydrogenase, is just going to do redox reactions. The polymerase is going to create polymers from monomers. Monomers is just a subunit, and polymers are just a bunch of subunits attached together. For example, DNA polymerase 3 is going to attach a bunch of nucleotides to create a long nucleic acid polymer. Phosphorylase is just the transfer of a phosphate group from inorganic phosphate. Let's say we have what it means to be inorganic phosphate, just to have like no carbon molecules attached to your phosphate. So remember, in ATP, the adenine, the nucleotide, is a ring of carbons. So that is an organic molecule. And an inorganic phosphate would just be a phosphate by its own, just a phosphate. And protease is a specific enzyme that cuts down protein peptide bonds. So just to go over the enzymes, right? The enzymes, what they do is that they are biological catalysts, which means that they're going to make sure that the reaction is going fast and not super duper slow, right? So although they help with the rate of the reaction, they're not going to change anything about the thermodynamics of a reaction. So let's draw the thermodynamics of just a random reaction. So this would be the thermodynamics of a reaction, right? The reactants have, let's say, 10 Gibbs, and then the products have 5 Gibbs. The net change would be -5 Gibbs, right? So that number is never going to change if you introduce an enzyme. The only thing that's going to change with the enzyme is that you're going to have a smaller activation energy, meaning like the activation energy was the wall that the reactants have to jump over in order to turn to products. Well, then the enzyme just decreases what that wall's height is, so the reactants can easily and quickly move on to the products, right? But the thermodynamics, aka how much product you're going to make, aka how far the reaction is going to go, is never going to be changed by a enzyme. Like if you were to make 100 units of something with this reaction, let's say in 1 day, well then your enzyme, it's not going to change that you're going to make 100 units. It's only going to change the time that it takes to make 100 units. So instead of 1 day, it would take 100 to make 100 units. It'd take 1 minute, right? An enzyme just makes the reaction go faster. So it influences the rate. It doesn't influence the outcome. And just a side note that reactions are for enzymes can happen in either direction depending on the situation. So an enzyme can, let's say, for example, just to explain that, an enzyme, for example, can catalyze the addition of A and B to make C, but the enzyme also can catalyze the separation of C to form A and B. So enzymes can go in either direction. They can catalyze reactions in either directions. But it really all depends on the exergonic or endergonic nature of the reaction. So remember, exergonic means that something is highly negative in Gibbs. So that means it's very spontaneous. And if it's endergonic, which means that something is highly positive or non-spontaneous. So if something is extremely spontaneous going forward, like let's say the ball rolling down the hill, like it's going to go down the hill, and then it's going to go here. That's something that's very exergonic, right? It's going to be releasing a lot of potential energy. Whereas the opposite reaction, the ball going up the hill, well, that's just not going to happen, right? Unless something's pushing it, right? So the enzyme, if it's something that's highly exergonic, it's not going to do the reverse step, right? Because there's not enough energy for it to happen. And so it depends on the situation, but sometimes enzymes can catalyze both directions. But sometimes if it's a highly exergonic or highly spontaneous reaction, the enzyme is not going to catalyze the reverse reaction. Okay. So important molecules, we discussed ATP, how it's adenine triphosphate. We remove a phosphate group, you get decrease in charge repulsion. Remember, phosphates have a negative charge. So if you have a phosphate group here, a phosphate group here, a phosphate group here, you remove one of those phosphates like such, and you have a lot of heat coming out. Well, that's going to be what we use ATPs for, right? These negative charge phosphates, they're repelling each other because of negative charges. So when one of them pops off, this molecule as a whole becomes more stable, because you have less electron repulsion. And also when you pop off that phosphate, you're going to gain resonance stabilization. Remember, if you go from highly unstable to stable, energy is released, and in this case of ATP, it's going to be released as in the form of heat. And that heat is going to be used to help catalyze other reactions that happen to be non-thermodynamic, right? That's reaction coupling. We talked about acetyl-CoA. So acetyl-CoA is just a group of activated acetyl group. So this is an acetyl group, right? We talk about how this is acetic acid. An acetyl group is just the carbonyl attached to a methyl, and then we have a something else here, right? We have something else, like that's an acetyl group, right? When we have acetyl-CoA, that something else is a coenzyme CoA. And what this does, what it is just a activated acetyl group, and this activated acetyl group is just able to enter the Krebs cycle, and in the Krebs cycle, we're going to be producing our NADHs and our FADH2s, right? Our electron carriers, and the electron carriers are going to be pushing the electrons to enter the electron transport chain and then create ATP. So that's what acetyl-CoA does. It helps us produce the NADHs and the FADHs that we need for the electron transport chain after the oxidation of glucose. Okay, next we have the electron transports, NADH and FADH2. So just to draw a quick ATC, say we had this right here, like such. Let's see, this is our ATC. We have the enzyme here. The enzyme is going to be the ATP synthase. ATP synthetase, which produces ATP. And let's draw our membrane here. You can remember the electron transport chain. If you guys remember from bio from high school, the electron transport chain occurs on the membrane of the inner membrane of the mitochondria. See, this is a mitochondria. This space here is the intermembrane space. This would be your outer membrane. This one would be your inner membrane, and inside here is your matrix. So when we draw this, we're just drawing this inner membrane, right? The ETC occurs in this inner membrane, aka the crista. So the NADH, it being the reduced thing it is, meaning reduced, it has what's it called, electrons. So the NADH, let's say for example, it just comes here, and it provides the electrons, so it's going to reduce the structure, right? If it's giving it electrons, it's reducing the structure. The electrons travel. Remember, the electrons have a negative charge, so we have a bunch of negative charges here, right? Just negative charge. Those negative charges are going to attract protons. The protons come here and pop up. And now the protons are like, hey, we have a bunch of protons here. We have a proton gradient. If you guys remember the osmosis, when we have like a bunch of stuff on one side of a membrane, and we have a little bit on the other side, what's going to happen is that the other molecules is going to passively go down its gradient and try to equalize the difference in concentration. So the protons that are pumped up by the electrons, what's it called, getting shuffled down through this ETC, the electron transport chain. Well, these protons are then going to go into this enzyme. It's going to cause the enzyme to churn, and if sufficient number of protons churn the enzyme, ATP is produced, right? The ADP and phosphate inside of the enzyme gets squeezed together, and you get an ATP molecule, right? And ATP is the energy currency of the cell, helping us with thermo unstable reactions, right? So that's what NADH and FADH do. FADH just introduces the electrons in a different spot. Insulin and glucagon, those are two. Those are two peptides from the pancreas. The insulin comes from the beta cells of the pancreas. Glucagon comes from the alpha cells of the pancreas. So what insulin and glucagon do is that they regulate the amount of sugar found in the blood of your body. So this is a blood. This is a vessel here. Is a blood vessel, and here we have a bunch of glucose because we just finished eating and we had some cake. And there's a bunch of glucose in our blood. Well, if there's a bunch of glucose in our blood, we're hyperglycemic, a lot high glycemic sugar. Well, that's not good, right? Having hyperglycemia is not good for your health. And so the insulin is going to be released, and any cell that's able to take up insulin, like a lot of cells can take up glucose. And for that reason, well, especially the liver and the muscle is able to really take up glucose, but mostly every cell in the body can take glucose. The insulin is just going to provide receptors on the surfaces of the cells so that the insulin can go in, right? Because we're trying to decrease the amount of glucose that we have in our blood because we're hyperglycemic. So insulin just decreases the amount of glucose that we have in our blood so we can maintain homeostasis, right? So that's what insulin does. It makes sure that we don't have so much sugar. And so to stop the amount of sugar that we have in our blood, it just tells the cells to pick up some sugar. However, let's say we're in a starved state, like we haven't eaten in a long time, so we don't have a lot of sugar in our blood. Then we're hypoglycemic. So to stop hypoglycemia, what we're going to do is we're going to have glucagon from our alpha cells of the pancreas innervate so it's what's going to do is we're going to interact with our what's it called? It's going to interact with our liver. The liver has the storage of glucose. The storage of glucose is called glycogen. So the glycogen molecule is really just a bunch of glucoses together. So what the glucagon does is it tells the glycogen to break itself apart so we can have a bunch of glucose. And then that glucose that's broken apart from the liver is going to be introduced into the blood. And we're going to increase the concentration of glucose in our blood. And we're going to prevent hypoglycemia, right, with glucagon. So these two are really opposites. If insulin is on, glucagon is off. If glucagon is on, insulin will be off. Okay, so quickly now let's talk about cellular respiration. So we talked about how cellular respiration is oxidizing our foodstuffs, and so we can produce ATP. Right, so we have 4 basic steps in cellular respiration. We have glycolysis, which produces 2 ATP. We have also a production of 2 NADHs, so electron carriers that are going to be moving the electrons from from whatever it is that we oxidized into the ETC. So the ETC will take those electrons and produce ATP. The pyruvate dehydrogenase complex, which produces 2 NADHs. The Krebs cycle, which produces a lot of NADHs, 2 FADHs, and 2 GTP. And for the sake of the MCAT, the GTP are going to be the same thing as an ATP, right? Just the difference of a nucleotide. Like in here, we have an adenine triphosphate, and here we have a guanine triphosphate. It's going to be the same energy levels, right? Okay, so for the ETC, we're going to, the electron transport chain, what we're going to have is just a bunch of the electron carriers like the NADH and the FADH just move their electrons into the ETC so that the ETC can produce the ATP that we need for our cell, right? So the products, the NADHs and the FADH products of our other steps are going to go to the ETC, so we can have ATP formed. So generally, just a rule of thumb for reactions is that, let's say we have a reaction like that goes from A through Z, right? We have a reaction that goes from A through Z, sorry, and there's like B, C, D, all those steps in between. Well, if we want to regulate our reaction, aka like either inhibit it or cause it to go faster, we want to regulate our reactions. It'd be smarter to regulate at the beginning, right? Because if we were to regulate at the end, well, then we'll have like a bunch of like reactions happen, a bunch of stuff that's a lot of reactants that's wasted, a lot of wasted energy. And at the end, we're like, never mind, let's like abort the mission. Let's not go to Z. So it'd be better to like in general regulate a reaction early on in the pathway. So that's something that we're going to find as a trend. It's not going to happen every time. Like sometimes we have regulations at the end of the pathway. However, it's just a rule of thumb that we tend to find mostly in pathway regulation. Okay, so let's talk about the energetics of glucose metabolism. So as we mentioned before, the NADH and the FADHs are going to be introducing their electrons to the ETC. Those electrons are going to be shuttled, and then the shuttling of the electrons are going to cause protons to go into the proton gradient, and then come back down. The enzyme that produces ATP come back down, churn the enzyme, and then cause ATP to be produced. Right, so that's how we get ATP from our electron carriers. So what we've talked about before is that the NADH, DH, and at the FADH2, they both carry electrons. However, they are going to donate the electrons to different components of this ETC. So the NADH is going to give it to component 1. The FADH2 is going to give it to component 2. So here we see that the electrons from the NADH are going to be having more time to interact with the ETC than the electrons that are given in the second component, right? So if it has more time, that means it has more opportunity to move protons into the proton gradient. And that's what we find. We find that the NADH actually moves 10 protons into the electron gradient, and that the FADHs actually move only 6 protons into the electron gradient. And what chemists have found is that it takes 4 protons in order for us to churn a ATP out from this enzyme. So it takes 4 protons to go 1, 2, 3, 4, and then produce an ATP. So if we want to see how many ATP that we can produce per of these electron carriers, what we would have to do is divide 10. So let's say we're talking about NADH, right? NADH, per NADH molecule, we get 10 protons moving into the gradient. So we divide 10 by 4 because it takes 4 protons to make an ATP. We use this ratio to find out how many ATP we can get per NADH. And what we find is that per NADH, we get, 2.5 ATP. So we get 2.5 ATP per, per NADH. And with FADH, we can do the same thing, right? We can take the 6 protons that the FADH shuffles up and divide that by 4 because it takes 4 protons to make a single ATP. We divide those two and we find that FADH produces 1.5, that's a 5, 1.5 ATPs. Right? So if we were to go back into this previous slide and we were to take all these things into consideration, let's just do this math real quick. So 2 ATP plus 2 NADHs, and remember NADH is 2.5 plus 2.5, that's 5, right? And 2 NADHs from the second system, so plus 5. 6 NADHs, well, that's 2.5 times 6, and that equals 15. 2 FADHs, that's 1.5 plus 1.5, that's 3. Remember, GTPH is going to be counted as ATP, so that's 2. And if we count all those up, we get a 32 in total, right? 2 plus 5 is 7, 7 plus 5 is 12, 12 plus 15 is 27, 27 plus 3 is 30, and then 30 plus 2 is 32, right? So theoretically, we should be getting 32 ATP from our all of our cellular respiration. However, what we find is that only prokaryotes actually get 32 ATP per glucose. Eukaryotes, like us humans, we only get 30 ATP per glucose. So what's the difference between prokaryotes and eukaryotes that can help us analyze why this happened? Well, prokaryotes, remember, they don't have any organelles. They don't have any compartmentalization. So what's going to happen is that they're going to have their NADHs and their FADHs just smoothly go over to the ETC because the ATC, remember we said before that the ATC is found in eukaryotes, is found in the mitochondria. However, for prokaryotes, it's just in their cell membrane, right? So they can just move and shuttle all of their NADHs and FADHs into the ETC and produce 32 ATP, no problem. However, for us eukaryotes, we have a compartmentalization, which means that we have to, if something is not in the mitochondria when we're doing the ETC, then we're going to have to shuttle it in there. And what we find is that glycolysis is the only step in this 4-step reaction that is not inside of the mitochondria. Rather, it's in the cytoplasm of the cell. So what we're going to have to do is shuttle these two NADHs into the ETC, into the mitochondria for the ETC to have access to those electrons, right? So what we find is that the glycerol phosphate shuttle, or the malate-aspartate shuttle. Malate-aspartate shuttle is just a bit more specific. It's just the glycerol phosphate shuttle that's in the liver, kidneys, and heart. And this shuttle, what we find is that when the NADH is being moved from the cytoplasm to the mitochondria, what we find is that the potency of the NADH to move electrons is the same as that of the FADH2. So it's as if those two cytoplasmic NADHs are going to be sending their electrons to this 2 compartment, meaning that they're only going to get 1.5 ATP per NADH if you're a cytoplasmic NADH and you're a eukaryote. So typically, eukaryotes only get 30 ATP and not 32. Okay, so glycolysis, okay, we talked about how this was a anaerobic oxidative catabolism of glucose. So let's break that down. Anaerobic means that it doesn't need oxygen for it to occur, which is awesome because glycolysis is somewhat of a universal cellular respiration pathway, because all living cells in all domains have the capacity to do glycolysis. So, that includes the cells who don't have oxygen around them, right? Because there's some cells at the bottom of the ocean that don't have any oxygen. So glycolysis is a reaction that can happen in any condition, aka anaerobic. Anaerobic means that there's oxygen is not required. Oxidative, we talked about how oxidative is just we're extracting electrons from something to move those electrons and use those electrons maybe for construction or maybe for ATP synthesis. Catabolism, remember catabolism is just the breaking down of things, of a glucose into 2 identical pyruvate molecules, right? So that's the catabolism, right? It's breaking down something big into 2 smaller things. Glucose, as we know, is a 6-carbon molecule. They're going to break it down into 2 3-carbon molecules, right? Those 3-carbon molecules are called pyruvate. And again, glycolysis is in the cytoplasm of the cell, and it's the first step for the cellular respiration cycle. So it takes 2 ATP to get the glycolysis pathway moving. Like we have to pay 2 ATP, but what we find is that we're going to produce at the end 4 ATP and also 2 NADHs. So if we add 4 ATP that we produce and then the 2 ATP that it costed us for glycolysis to happen, if we do 4 minus 2, then we get a net production of ATP at 2, which means that the net production of the glycolysis pathway is 2 ATP, right? Because we made 4, but it costed 2. So 4 minus 2 is 2. We're going to have 2 pyruvate and then 2 NADHs. So here at the bottom of my notes, I have specific kind of like niche things like GLUT4 receptors and fructose and galactose. Just really just go over it on your own. They're just like specific things that you might find. Okay, so really quickly, I'm going to show you guys a little bit about the pathway for glycolysis. So let me pull up this so you guys can see this pathway. So we talked about how really what you need for pathways is to be able to interpret what the enzyme is saying. The name of the enzyme is also the name of the reactant in the product. So here we have a glucose molecule. We said that there's just a Hartworth projection, and for glycolysis, we're going to have the glucose molecule in either the Hartworth projection or in the Fischer projection, depending on what it's doing. So you have a typical glucose. We're going to have a ATP donate a phosphate to the glucose and form glucose 6-phosphate, right? So that's an aptly named name, right? It's a glucose molecule with a phosphate on the 6th carbon. And we're going to have this reaction be catalyzed by hexokinase. So one of the students, Julia, she was just like, oh, does the hexo mean that it's just going to lie on the 6th carbon? Well, I looked it up and I have the definition. I have it on my phone. I'm going to read it out loud. It is, let me see here, I have it on Chrome. As a hexokinase is an enzyme that phosphorylates hexoses, aka 6-carbon sugars, forming hexose phosphate. So it doesn't, the hexo and hexokinase doesn't specify that it's going to be adding it onto the 6th carbon, simply that it's adding a phosphate group to a 6 carbon molecule, right? So it's a hexokinase. And then this step right here is going to be an irreversible step, simply because it's very exergonic, right? This breaking up, we said that breaking up of ATP is going to release a lot of energy. So moving this backward is really not going to, it's just not going to happen. And we talked about how having a phosphate group on the glucose is going to trap the glucose into the cell, simply because of the charge on the phosphate. It's going to prevent it from moving out from the cell membrane, right? So the plasma membrane is going to keep this thing stuck in here, because the plasma membrane inside we have the fatty acid tails in the bilayer, and that fatty acid tails are non-polar, right? So they're not going to want to be interacting with this charge. So the glucose 6-phosphate is now trapped inside of the cell. And we talked about this about these reactions in lecture. So you really should have been should have really gone towards lecture, so we can really just go over these reactions. I don't want to go over all of them, because that would just make this quick, like, kind of like summary video too long. But just to show the most important ones. For example, this one, the rate limiting step, is just the step that it really is the slowest of all of the steps in the what's it called, in the pathway, which makes it the most important step in terms of determining rate, right? So here we have a fructose 6-phosphate, aptly named because there's phosphate on the 6th carbon of the glucose. We're going to have another ATP sacrificed, and it's going to put another phosphate. And the enzyme that catalyzes it, phosphofructokinase, very aptly named a phosphofructose, because this is a fructose fructose, and we're going to add a kinase to it, right? So that's perfect, right? Adding a kinase, adding a phosphate to a fructose, right? Phosphofructokinase. And again, our very aptly named product, fructose 1,6. So there's a carbon on the 1 group. So there's a phosphate on the 1 group, phosphate on the 6 group. Bis, so bis is bi for 2, bis phosphate, right? Aldolase, remember talking about the aldol reaction. Aldol reactions, 2 carbonyls just attack each other. In this case, these are the carbonyl here in this ketone, this carbonyl here in this aldehyde. They're going to attack each other and form this fructose 1,6 bisphosphate. But in this case, remember we said that enzymes can be reversible. So in glucose glycolysis, we're going to be having the forward direction of the aldolase going in this direction. In gluconeogenesis, we're going to be having the backwards reaction, right? Which is combining those two, molecules to form the fructose 1,6 bisphosphate. And just to summarize, where you have Hartwig and Fischer projections. So whenever we have a Hartwig projection, we're able to add on a phosphate group. Whenever we have a Fischer projection, we're going to have a different type of reaction. We're not going to be adding phosphate groups. We're going to like maybe isomerize something. Like here we have a phosphoglucose isomerase. There's phosphoglucose right here. Like there's an aldehyde here, and then the isomer have a ketone on the 2nd carbon, and the aldehyde is no longer so the aldehyde here was on carbon 1, but here on carbon 1 we only have a OH group. And what happened is just that the aldehyde here was reduced into an alcohol group, and that the 2nd carbon oxygen alcohol group was oxidized into a ketone. And then it went back into the Hartwig to get the ATP, and then it went back into the Fischer for it to be cut by aldolase. And I talked about how knowing the structures are very important. There's a glacer aldehyde 3-phosphate. So we talked about the glycerol group. The glycerol group is just a 3-carbon group with each carbon having an a alcohol group. And aldehyde means that one of the groups at the end has to be a carbonyl group that has a H poking out. And then 3-phosphate is just the location of the phosphate group, right? And then we talk about the dihydroxyacetone. Acetone is a group which we have a carbonyl and then 2 methyls sticking out as the arms. That's a acetone. When we have a dihydroxyacetone, I mean di means 2 and hydroxy means a OH group. So there's going to be 2 OH groups, one on either end of your acetone, and then one of those OHs is going to be replaced by a phosphate. Remember, the phosphate is just going to pluck off a hydrogen from the alcohol group, from the OH group, and is going to bind to the oxygen, right? So we talked and continue further. Can see if there's any other important ones. I just remember the names of these reactions, right? Just like an enolase reaction. An enol we talked about. An ene is a double bond. A O is just indicating that there is a alcohol next to the double bond. So as we can see here, we have an enolase form a enol group right here. It's a double bond, and next to it we have a alcohol, except the hydrogen on the alcohol is replaced by a phosphate, and that's why we have a phosphoenol, right? Yep, so then we have our last step that's irreversible, is our pyruvate kinase. We're going to make our last substrate level phosphorylation. Substrate level phosphorylation is when we have a molecule itself, add the phosphate onto the ADP to make ATP. And so this is our glycolysis pathway. Next, let's talk about like there's the irreversible steps I have listed here on my notes. So just we have like these reversible steps here. Like hexokinase was irreversible, PFK was irreversible. Also the commitment step, and then step in the third irreversible step was the pyruvate kinase, the last enzyme. And just remember that certain things can inhibit enzymes, like hexokinase is inhibited by G6P. Fructose phosphofructokinase is inhibited by ATP, low pH citrate from Krebs, but it's positively regulated or made to go faster by AMP, phosphofructokinase, and fructose 2,6-bisphosphate. So just to talk about a little bit the AMP and the ATP, so just recognize that if we're doing the catabolism of glucose, our main goal is to produce ATP, right? But if a cell is full of ATP, then we don't really need any more ATP. So if there's a bunch of ATP in the cell, it's going to tell the cycle or the pathway like, okay, we don't need like glycolysis, you can like chill out, right? So that's why in phosphofructokinase, ATP is inhibiting it. AMP, however, adenine monophosphate, which means that the adenine triphosphate has been dephosphorylated twice. That means we don't have a lot of energy in our cell, like we don't have a lot of ATP, which means that the AMP is going to positively regulate or make the reaction go, just to make sure that we from glycolysis that we get ATP needed by the cell. Okay, next we talked about the pyruvate dehydrogenase complex. In the pyruvate dehydrogenase complex, what we have is the pyruvate from our glycolysis reaction. Sorry, just needed to wet my whistle there. the pyruvate from the glycolysis pathway is going to be oxidized. And remember, if we're oxidizing something, we're extracting electrons. Is going to be oxidized within the mitochondrial matrix. So the pyruvate is going to travel from the cytoplasm, it's going to enter the mitochondrial matrix. The pyruvate is going to be oxidized, and at the end of this reaction, we're going to create the acetyl group. Remember, the acetyl group is going to be the thing that enters the Krebs cycle to produce all those NADHs and all of those FADHs. So let's look at the mechanism. Here we're going to have our pyruvate group. The bond here, remember, we're going to oxidize it. So if we oxidize it, we're going to increase bonds to oxygen. So this sigma bond between those two carbons is going to go to this oxygen group here. We're going to have a decarboxylation, an oxidative decarboxylation. And what we're going to be left with is as you can see here, a carbonyl group and a methyl group attached to it, right? So this is your acetyl group. And then the enzyme is just going to attach a coenzyme A, just to activate this acetyl group so it can go into the Krebs cycle, right? So here's the reaction: the pyruvate, an oxidative, decarboxylation. The CoA is attached to your to the acetyl group. And remember, if we have an oxidation, that means that something else is reduced. We don't want those electrons that were released to be going to waste. So our NADH is going to catch those electrons and be reduced to NAD. Our NAD plus is going to catch those electrons and be produced into an NADH. And remember, our CO2 is the thing that's released, right? And how do you know if an NADH is going to be potentially used? Look at the name, right? Dehydrogenase. Typically, when we have a dehydrogenase, it's going to be a redox reaction, and we're going to be using an NADH or an FADH. And then last, we have our acetyl-CoA group, right? And down here we have the what it's inhibited by and what it is positively regulated by, right? And I believe we ended our discussion here. So I'll be seeing you guys in the next video.